Hello, my name is Radha Noor, and I'm in the 11th grade, and I'm going to be discussing the neurological impact of diabetes and how stem cell could be a potential course of action in humans. And so first I'm going to start by explaining some background information about the condition diabetes. And so first of all, diabetes is related to how the body manages blood sugar level. And so insulin is one hormone that is used in order to do this and it lowers blood sugar level. And it's produced by the pancreas through the beta cells that respond to higher or lower glucose levels and also the smell of food. And so, specifically type 1 diabetes is related to how the body can't produce insulin since the beta cells have been destroyed. And while type 2 diabetes is more related to insulin resistance and beta cell strain due to that resistance. And some other key terms that are important to know for the method of presentation is hypoglycemia, which is lower blood sugar level, and hypoglycemia, which is high blood sugar level. Now, so throughout this research program, I was mainly looking to investigate specifically what the impact of diabetes was on neurodegeneration and how this could either be prevented or cured. And so some questions that I had that I wanted to answer throughout this was, first of all, specifically, what is the impact of diabetes? And then second of all, some research found that there was these FJB plus biomarkers that were found in hypoglycemic conditions. And this was in a 2009 study by Lee et al. And I wanted to also look at how stem cells could be used as a cure for diabetes to prevent any complications from arising. And this is an important area of research since the diabetic population is ever growing and projection numbers are expected to continue to grow up into the 2030s, 2040s. And so far, it's not showing much sign of slowing down or stopping. And so there was multiple research that was conducted, such as on hypoglycemia or the low blood sugar condition. And one potential reason for the FJB plus biomarkers was glutamate excitotoxicity, or stimulation due to excess glutamate in the synapses. And this results in the death of neuron cells. Neuron is a protein in the nucleus of some neurons. And the death of these cells results in the formation of FJB plus biomarkers. In addition, in the Brie et al. Study, there was 2 times the number of these biomarkers in diabetic rats compared to non-diabetic ones. And they hypothesized that insulin deficiency in the brain was the cause of this. And research shows that this might be the case as insulin deficiency results in amyloid beta formation, amyloid beta which is related to Alzheimer's. Hypoglycemia was also studied, and it can cause nerve damage or diabetic neuropathy through formation of age or advanced glycation end products, which when they are accepted by RAGE or receptors of age, it results in inflammation. In addition, excess glucose results in the formation of acetyl-CoA inside of Schwann cells, which are used for the formation of myelin sheath. And excess acetyl-CoA is through a process is then converted to toxic acylcarnitines, which result in mitochondrial dysfunction. Stem cell was also researched, and there was some success in using stem cell to cure diabetes, such as in a 2024 study, and where a type 2 diabetic patient was cured using stem cells. But this may not exactly be the case, as stem cells, especially for diabetes, have been shown to degenerate over time. And one solution for neuropathy was also looked into called TMS, or transcranial magnetic stimulation, which is a non-invasive technique that uses repetitive magnetic stimulation in order to handle the inflammation caused by neuropathy. And this has been shown through some research that this could be a promising technique. So the study used only online sources, no physical sources, and the study found that looked at many things such as how cortisol could be a factor leading to diabetes, how glutamate excitotoxicity could lead to neuronal death, how AIDS and acylcarnitines cause neuropathy, the benefits and drawbacks of stem cell and emerging methods such as TMS and how they can be used to help patients with neuropathy. And so the study was really looking at how the impact of diabetes and neurodegeneration and what's in potential cure as well. And information gathered on the hypo and hypoglycemia, stem cell TMS, and other processes and methods that were involved with how they progressed and everything. And so research on stem cell and TMS showed them to be both as promising ways, but further research needs to be done in order to help define both of those processes. The study also helped to expand upon the Lee et al. Study, which was focused on the FKB plus biomarker, and helped to add on that about their hypothesis of insulin deficiency. It also helped to provide another opinion for the 2024 case in curing diabetes using stem cells, and highlighting that potentially more research needs to be done since stem cells can degenerate. Now, some constraint in terms of this study was that there was not enough time to completely go into full detail and everything, and the scope was a little bit broad. But overall, there was a good general review of everything was able to be provided. My experience with this research project was that I had a really good experience with working with all the professors. For example, Professor Walke helped me learn about TMS, and without his advice, I would have never went into detail about that method. And then Professor Wojcik also helped me to organize my paper, and without any of the help, my paper would have been completely different from how it is like now. And so throughout this entire process with the company and everything, I was able to explore a topic that I was interested in and just learn more about the topic in general. Thank you.
Good. Question for Raza. You can turn off your presentation.
A good presentation, and I can see that you have put effort in trying to understand. Although this is a broad topic, you try to work on something very specific. It's good that you know a little bit more in detail about what goes in type 2, type 1 diabetes and all. I have a question. So do you know how does TMS work?
Yes, so I did some research onto how TMS works a little bit. And so some research I found was magnetic stimulation impacted this BDNF PLK-DA pathway. And so through that, these are different types of so through impacting this pathway is I don't exactly remember, but
I mean, is it like related to the, what I mean to ask is rather than the specific pathway, in general, how does it affect? Like, what does it do?
In general, like the research that we were looking at before was kind of explaining how TMS could be used to work against inflammation inside of the body caused by neuropathy. So neuropathy results in inflammation, and they explain how TMS could be used to kind of reverse that inflammation and reduce the stress that was caused by high blood sugar condition.
Okay, by stimulating the neuron.
Yes.
Yeah. Okay. Okay. Good job, Raza. Hope that you continue to expand upon this research too, and also best wishes for you. Let me know if you have any other question anytime.
Thank you.
Okay.
A big round of applause to Raza for his wonderful presentation. Next up.